Someone's wondering, what are some of the features that Herc is specially equipped with for this dive? Were there any particular instrumentations specially attached, Jess?
That's pretty standard this dive. No, nothing crazy. Every dive is special, but nothing too outstanding that we put on the vehicle for this one.
Yeah, on the starboard manipulator, we have the parallel jaws on the manipulator. And our intent for selecting those, as opposed to the ones that have kind of interlocking fingers, was that we wanted to be able to pick up rocks and try to squish them and see if they would break. And that would give us an indication of whether they were altered or not. And you can have a little bit more grip strength with those than with the interlocking finger type manipulators. And the other, we put on the front basket a scoop so that in case there were rocks that were too big to fit in the manipulator pads, that we could try to pick it up with the scoop. That's as technical as we got on this dive.
Exciting. I have to ask, what is the maximum pressure that the
Jeff, you want to go ahead and push in
manipulator can force?
While we're waiting.
All right, Steve, what are we looking at here?
We are looking at a Chrysogorgia octocoral, the golden coral. It looks like it belongs to the same genus and species we've been seeing for a while. This is Remulogorgia, probably Remulogorgia militaris. It is it's one of a few of the common species of colonies that look like this in the area.
Okay, go ahead and come wide there, Jeff.
When we've seen these throughout the dive, I don't think we've seen much indication of other animals using the coral body, either for lift or for predation. Mm-hmm. Is that common?
Yeah, it's associate organisms generally follow the same rule as a lot of the other megafauna. So the deeper you are, the less you'll see. Mm-hmm. There are some species of corals that across the entire depth range they occur. You'll find the same exact species of associate across their entire depth range as well. This is quite common in some other Chrysogorgiid species. But in this case, I've only seen a couple
of
brittle stars associated with these Pleurogorgia colonies.
Some cool sponges here.
Yeah, what is that?
Yeah, what?
There's a fish there in the center.
Yeah, there's a fish.
Something, is that sediment or some sort of encrusting? Odd little pocket of sediment there.
I think it's a little, yeah.
I'm not sure what
ground.
Oh, it's like it looks like a sheet flow with some sediment on top.
Is that sediment there?
Or detritus, whatever.
And what is that?
Is that an acorn?
Purple thing on the left?
Yeah, I think
Oh, an acorn worm? Yeah.
Yeah, well, you come back to that. I'm not sure what that is.
Or a holothurian.
Yeah, it's probably just a flat part of like a sheet flow that's got a little bit of sediment dusting on it. Yeah, and then we got this little fun.
Maybe it's just a holothurian. Not just a holothurian.
I think that's our second holothurian sighting for the dive, right?
Yep, that's number 2.
Do a partial there real quick.
Looks like it's got interesting stuff going on the inside there.
Recently eaten.
Yeah, it looks to be a second species of sea cucumber that we have seen on the dive today. The first one we saw was Anipniastes, and this one looks to be a little bit different. Could be Benthodites, but I have to double check that.
See, it's little tube feet on the bom.
I think doesn't look like if it is, it does, yep, they might actually lift off there. There's a lot of different types of these swimming sea cucumbers, but they're not entirely pelagic. They might just swim a little bit and then settle back down on the seafloor. But if perturbed enough, that they would definitely lift
All right,
off.
go ahead and come right there, please.
Most sea cucumbers are detritivores, right? So kind of moving along the ground, cleaning up after anything else.
Yeah, exactly. They'll pick up some of the particle debris, try to ingest whatever's nutritious, and leave the rest behind.
Definitely seems like there's a lot of dusting or detritus here.
Yeah, so this area doesn't look as current swept as some of the other spots that we've been in.
So before I call in another move, or any of these rocks or anything looking sampleable
looks
to
like
you?
something.
Yeah, good question.
No pressure.
Yeah, right? A lot of these look attached. What might be good? Can we maybe see what these look like here?
Sure.
Don't know if we have enough time to slow down and have a look.
I think we're okay.
Yeah, we're all good.
Now that we're getting closer, I'm seeing that that looks attached. I thought maybe this one was loose, but now that I'm seeing it closer up, it looks like it's welded in there.
Should we keep on checking then?
To the left of the lasers, that looks a darker one.
Yeah, I was looking at this,
Yeah.
but it doesn't. From further away, it looked like it was unattached.
Yeah.
Do you want to do a partial there, Jeff?
Sure.
That's great. Is that a negative on this guy?
I'm wondering if either of these would also be good for Andrea. They're nice and angular.
Oh, Raj.
Can always poke at it.
Yeah. Pull wide there, please.
So again, we're kind of targeting two different kinds of rocks here. This one would be an example of a unaltered or hopefully less altered rock.
Jeff, go ahead and push on in.
Maybe. So the ones right just to the right of the laser marks, which are nice and more angular, might be decent for some of the age work. It's hard to tell from this view. Definitely got some kind of, I don't know if that's a nematode or something attached to it. And then off to the right, outside of the field of view, there's some that look a little more highly altered. I don't know. These look like Might want
Did
to.
you mean to the left or no?
There's also one to the left. Yes, you're right. But there's also one out of the field of view.
All right, go ahead and come wide there, please.
So I'm just curious if, especially this one, might be able to be picked up.
Do a partial there. Better grip on this guy.
Well, they both are pretty loose. All right. Can we have a zoom in on that piece that we just picked up?
Sure thing. Do you want me to rotate it around, or
Yes,
would
please,
that
if you could
disturb this?
rotate it.
Great.
Almost looks like a charcoal briquette.
A little picnic on it or something.
Oh,
Yeah.
kind of little sea
Oh,
spider.
little sea spider.
Could we, I don't know what your grip strength is right now. If you could try to give it a little squeeze and see if it feels robust or if it's just going to crumble apart.
Sure thing. Yeah, it was a grip force of 7, so let's try a little more.
Yeah,
That
it
is
seems like it's holding pretty well.
full grip.
Okay, especially since there's two pieces that are similar here. It might be, I think it's worthwhile to take both of these pieces and put them in the same compartment in the starboard boxes.
Sure thing. Pull wide there, please. And what do we got open in the starboard bio?
We have A, B, and E. So that's the two inboard and most forward, and then the E is the outside forward box.
ABE, roger.
So is this
Basically, the forward half of the box is all open.
Steve, would this be sample 7?
This will be sample 7.
Okay.
Yeah.
Switching cameras.
Thanks, Jake.
We hope with these stronger rocks that they're representative of some of the more original material here.
Hopefully they have preserved, yes,
Right,
some of the original material. They're not too highly altered so that the original minerals can be separated apart to try to target particular minerals that kind of hold a clock for the age of the rocks.
Forward.
Yeah, sorry. So Steve, what were the?
Yeah, so E is the largest compartment is open, and then the two inboard from that are available as well. So A and
Go ahead and keep popping it out then, please, Jake.
the whole front half of the box is open.
Front half.
Yeah.
Well, then let's go for B. Oh, we're putting 2 of these guys in.
Yeah, the two so the one that you're holding and the one that was right next to it, we'll put them together.
I think they can both fit in B.
Does it make sense to call them the same sample?
I think so.
Okay.
Yeah.
All right.
They're both coming off the same structure and they look very similar.
Mm-hmm. So that's going to go into compartment B.
Slow moving box.
All right, seeing that sample box close
Roger
back
that.
in.
Thanks, Jake. Okay, and then we're going to go for this other angular bit right next to it.
Correct. It's twin.
Do you want to zoom in before I poke it?
Yes, please.
Okay.
All right.
Great. You want to do partial wide there, please? Wonderful. Oh, terribly good. All right, you want to push that in there, please, Jeff?
All right, what was that?
Yeah, so this one's looks very similar. Another little charcoal briquette. All right. Thank you for that zoom in. I think
Sure
we
thing.
can go ahead and put that in the same compartment B, please.
Sure thing. Full wide, please.
All right, switching.
Sure thing.
We've got Jeff over here switching the cameras that we can see on our quad view. You can watch the whole process as Jess turns the manipulator arm and deposits that rock sample with its friend.
Beautiful.
All right. And then Jess, if we could, just see if this piece is loose.
Sure thing.
And then if it is, we'll hold it up and see if it looks like it is any more altered than what we just picked up.
Go ahead and push that in there a little bit, please. This big old one. This is the one we're looking at? This guy here?
Right here.
All
Yeah,
right.
if you kind of like touch it with the manipulators, does it seem like it's loose?
It looks loose, yeah.
It does look loose.
Cause a little avalanche here.
Jeff, do you mind if we get a zoom in on that rock? See if it's worth picking up.
Let me see if I can get you a better angle.
Yeah, better position a little bit here.
Yeah, we're as full wide or full to the right as we can get.
Okay, right.
If you want, I can turn the vehicle.
No, it's okay. I think it looks to me like that's gonna not be good for what I need.
It looks pretty big too.
It's pretty big, but also it looks like it's not really altered. It's not giving you any give when you touch on it.
No, yeah.
So yeah, I think we'll say we've got what we needed in this spot. Say thank you to this spot for those nice rocks that didn't require too much work. And we'll keep moving on up.
Roger that.
Mahalo.
I don't know if you've already called in that ship move or if we still need to do that.
Yeah, I'm just we'll wait till we get stowed and out ahead. Or I guess we look scarier than it is, but it's not so bad.
Yeah.
I've been like looking
Still 10, 15 meters off, which is good.
Yeah.
So
But
hopefully,
yeah, it might take a little bit of time, so I could call it in now if you're all right with that, Jess.
yeah, go ahead. Hopefully those rocks will give us some information about the age of the seamount, which ties very nicely with one of our audience's questions. It's almost like there's a plant out there. Somebody's wondering, are these seamounts formed by the same hotspot that formed the Hawaiian chain? If so, how come they have a sort of southeastern angle?
Yeah, that's a fun question. So for those following along at home, if you have access to Google Earth or any other kind of program that might help you see these seamounts where we are, in relation to the trajectory of the Hawaiian island chain, you would see that, so the Hawaiian chain kind of goes from a west-northwest to a east-southeast trajectory. And the seamounts that we're looking at are a small chain that's more of a northwest-southeast orientation, where if you were to draw a line, they would kind of be oblique to one another. And it's a very hot topic of discussion in the field of marine geology to try to understand what the potential origin is of these seamounts, and if they are contemporaneous with the Hawaiian island chain, or if potentially they're older than the Hawaiian chain and maybe had a different origin. And so that's actually why we want to try to collect these samples to try to understand that and try to understand the history of the formation of these features both within the monument as well as how the features within the monument connect outside of the monument.
And we're talking a difference potentially here of like 50 to 70 million years. Is that right?
Could be. So the Hawaiian island chain, the current the island of Hawaii, the big island where there are active volcanoes, right? That has roughly a 0 million year age. It's still being formed. And as you move out to the west, those formations are, let's say, in the 30 to 40 million year age range. And then as you keep going towards the emperor seamount chains, they get older and older. It's possible that these seamounts that we're studying and others in the Papahanaumokuakea Marine National Monument that may have been formed, yes, you're right, tens of millions of years or more different from the Hawaiian Islands.
I've been thinking about this a little bit, like if you have a piece of fabric on a sewing machine and you move it through, you see that thread pattern, and that's a little bit like how you see the pattern of island formation.
Mm-hmm.
So the question here is, was that thread kind of an offshoot of the same one, or did it actually cross over at a different time completely?
Yeah, Dr. Chris Kelly, who is one of the other co-lead of this expedition, in his presentation gave a great analogy of imagining footprints and tracks on a beach, and how you might have one set formed and then somebody else comes and walks over that in a different direction. And so you can kind of see by the layering which came first, which came second, and that you know, one might have been in one direction, one might have been in a different direction. So again, we're seeing some slightly different features, rock features, as we're moving up. So we had been seeing a bunch of pillow basalts. Now we're seeing things that look more like sheet flow. So these are going to be very hard to try to sample. But again, in that we're in a spot here that doesn't seem to maybe have a great amount of current and marine snow. There's not a lot of attached life on these rocks compared to some of the other features we've been visiting.
Well, you sparked a bunch of rock puns here in the chatter.
Oh, really?
With your hot topic comment.
Okay.
Hot topic and
Haha.
lava. Haha.
Yeah. I wish I could say that those puns were intentional, but I'm not that clever. So again, we're seeing some evidence of decaying and dead sponges, but not a lot of live sponges in this spot.
You know, on a different note from kind of the scientific geologic record, I'm wondering, Kalamehu, if you could speak to some of the more cultural or even just linguistic genealogy record that exists within the Hawaiian culture about this region.
That's just about like Papahanaumokuakea in general.
Yeah, and how that record has been kind of orally passed down with.
Oh, okay. Yeah, so from what I understand about Papahanaumokuakea in my limited capacity is that, you know, due to extensive ethnohistorical research and stuff like that, particularly within the regions within the Nihoa and Mokumanomana regions, they are actually recorded within other historical chants and in primary Hawaiian language resources. Since then, I think it's been a long collaborative process with the Cultural Working Group and other hui such as the Edith Kanaka-Ole Foundation and various communities across Hawaii to sort of discuss the cultural history and also the appropriate sort of means of moving forward in terms of like the nomenclature of these islands. Although, well, within like the greater sort of like repositories of like Hawaiian of like cultural stories and stuff like that, we do have there are mentions of places such as Mokumanomana or Kue Helani and stuff like that, and in the stories these are actually the birthplaces of the like the ancestors of some of our older gods and chiefs like that. And so for instance, one mo'olelo that I can or story that I can think of right now is named Kianini Ulo Kalani. And he's a chief that his ancestors are actually born in Kuehelani in Papahanaumokuakea, and a few generations later he actually he sails down the island chain all the way down to the big island of Waipio, and he lands at Waipio to find a wahine and stuff like that, or like a wife, if you will. But then otherwise he's making treks back up in there and stuff, and then the story jumps back between what's happening in that region and then sort of what's happening. So jumping back and forth, but otherwise just to sort of zoom out a little bit on that. But some of the things that have been shared with me are that like generally looking at like the Hawaiian archipelago and stuff like that, and in like Hawaiian thinking and the view of the Hawaiian world, we usually would think that like life will begin in the east and stuff like that and will set in the west, therefore following the path of the sun. And so in that sense, and then like what was shared earlier, the Tropic of Cancer, which was where Mokumanomana actually falls upon, they see that as sort of like a boundary between the more secular world in which we all sort of reside in, the Al Kanaka, which is gonna be where humans live and stuff like that. Sorry, but and then up past that is gonna be Po, or basically like literally night or darkness, but more figuratively. So this is a place where our more deified ancestors and stuff like that would reside. And so like it's almost like if you think about not only their geological ages and stuff like that in succession, but when you think about it more so as like this is an opportunity, and this is the sorry, I'm kind of talking donuts right now, but anyways, yeah, sorry guys.
No.
I think it's fascinating how it aligns with the geologic history as well.
Yeah, for sure. And so like, and then in that we have these ancestral stories that are based in Papahanaumokuakea. I think that only adds to the added cultural value of these areas as well. And then I just wanted to bring up too, like, so just as we have like the science and operational and safety protocols, we also do have the cultural protocols as well that we've been following on this voyage. If any of our viewers and stuff like that are interested in looking at and examining these protocols and other the cultural history of Papahanaumokuakea, I feel like I'm not doing the best job right now. you guys can go to the Office of Hawaiian Affairs website and look at the Papahana Mokuakea Monument. And there you'll find some other features such as like some of the history of the conservation and protection of the monument, as well as some of the stories that I made a mention, as well as the protocols that we're employing here as well. So I would strongly encourage our viewers out there to go take a look at these things just to give yourselves a little more context about just where we are in like our culture history. So mahalo for the question.
Mahalo.
Thank you.
I echo that. You can find a link to our particular expedition, and from there you can link to some of those resources that Kalamaiu just referenced.
So it looks like we have another maybe stocked sponge in the view here. A single solitary.
Yep.
It's over there. All right, just turned my microphone around, so please let me know if it sounds okay.
Just a heads up for the science team. I think we are on camera currently on the quad.
Yes, we are.
You actually have been for a while.
Yeah, I know.
Oh, the eclipse?
Oh, is it? Oh, all right. All right. Looks like we're maybe viewing a tailless field here, a lot of rubble, as well as these big blocks. Just the extent of the rubble is maybe a little more apparent in the view from Argus.
Yes, we are. I called a ship move in before I left, so I think we are. Yeah, probably got another 25 or 30 meters. Argus may have slowed down there for a little bit.
Rennie, do you mind zooming out on HIPAC so I
Sure.
can see where we are in relation to the presumed endpoint? Thank you.
And that is 750 meters away.
Okay.
If you like, I can, rather than calling in these steps, which are at a speed of a half a knot, I could do a continuous move at 0.3 knots, so we can.
Oh, the end target is somewhat arbitrary.
Arbitrary,
It's nice
okay.
to have these steps so that if we want to sit down and do some sampling, it's a little bit easier.
Sure thing, we can keep going as is.
All right. Take a moment here.
Another sponge tucked up in there. From the start of the dive, yeah. I want to say half. Yep, we've gone about 650, and we got 750 to go.
We're having a few questions about kind of the logistics of our watches. And if you're tuning in here, you can see everyone who's currently in the control van underneath the quad view on the main Nautilus live page. So if you're hearing my voice, just scroll down, you can probably see everyone. I'm Lindsay, your science communication fellow for this watch, and our watch is from 8 to 12 Hawaii Standard Time. We do rotate watches every 4 hours, regardless of, for the most part, regardless of where the ROVs are at. And we do tend to set dive times or estimated times. Of course, that's always able to be flexible and change. I think that this current dive was originally estimated to be about 12 hours. Is that right, Beth? Mm-hmm. We're getting close to halfway through that.
Oh.
Seeing another one of those bamboo corals. We've seen quite a number of these at this point in the dive. That characteristic bend about 1 meter up. We do have the ability and permission to sample some of these corals and sponges. However, we would prefer to do that in limited amounts. Really, the primary objectives on this dive are geologic. So I don't think getting to the very pinnacle of the seamount was part of the dive plan. Is there a reason that we're not interested in going to the very peak?
Yeah, good question. So I was mentioning earlier, how do we pick our dive targets? And so one of the goals is to try to understand the depth distribution of some of these coral and sponge species, and in particular to understand their kind of maximum depth distributions. And so prior work by our colleague Chris Kelly, who's part of this expedition as well as others, suggests that maybe there's a sweet spot between 2,000 and 2,500 meters water depth to try to figure that out. And so that's why we picked this region. Now there's a fun brittle star bamboo coral combo there in the frame. And so we picked a depth a little bit deeper than that to start the dive, just because of the feature, a scallop feature in the multi-beam, indicating some mass wasting. But getting to the top of the summit wasn't a priority to get all the way up to about 1,300 meters, because it was thought that the coral and sponge communities would be similar to what had already been seen.
Thank you.
Nice big boulders here on this part. You were right, your comment earlier, Steve, about you're more likely to find some of these species on the bigger boulders that maybe have less likelihood of moving around. Seems to be holding true.
Yeah.
And remind
Yeah, and you get, the other component was you get topographically induced flows around some of these larger boulders where currents will speed up, and faster currents usually signify faster food transport in that area, so it's advantageous for corals to settle in those places.
Definitely advantageous for that brittle star.
And also, yep.
So we have a question for you, Beth, about how you're selecting your rocks, and the viewer's concerned that it's a bit like finding a needle in a haystack, but how concerned are you about the optimal location of your rock sample? Is that your primary concern?
Yeah, right, because you see this field of view and there's hundreds of rocks. How do you know which ones to pick? And partially we will pick many different samples throughout this dive at different depths to kind of give ourselves an idea of the variability in terms of what the ages might be, because it's not as if this entire mountain was formed all in one day. Could have been formed over millennia. And so we want to pick up rocks in different places and have some randomness to that. And so often our limiting factor is just, can we pick this piece of material up? And then trying to see if visually it indicates that it might have the features that we want. But there's always a little bit of a gamble. What you see on the seafloor versus what you see when it's recovered in the laboratory, sometimes you didn't quite get what you wanted. And so there's always that trade-off of, we don't want to collect too many things, but we also need to collect enough to kind of give us some hedge against not getting our target.
Thank you. Helpful.
And with some of the analyses that are proposed for doing these measurements, you don't necessarily need a lot of material as long as it's quality material. So even though the sample size may be small, it's still maybe good enough to get you what you need and to have actually still even enough material to do replicate analyses. Rennie, how are we doing with the current ship move in terms of, is the ship where you wanted it to be yet? Are we still, is it still
yeah,
moving?
yeah, it's a bit of a delay. So
Okay.
I just called one in. The ship is moving, and Argus will follow suit.
Okay.
And yeah, so I just called one in for 30 meters. I would expect Argus. Argus still had a little bit of movement from the last one, and just kind of like layering them. There's a bit of a speed up and a slow down.
Okay.
Is that okay?
Maybe at our next little
Mm-hmm.
hop, we'll see what the rocks look like down there.
Yeah, certainly. So
see
we'll
if there's
just,
something else worth picking up.
yeah, we'll I'll keep track of the 30 meters there, and we'll hold at the next one.
Like there's like a largish sponge or something directly ahead, right?
Yeah, potentially. Water seems more turbid where we are right now.
So just that ship is just about complete its move. Argus will have about 30 meters, and then we're gonna take a look at rocks. Oh, that was a big stalk.
Yeah,
Yeah.
really
What was that, Steve?
quite thick.
That
Big
one
one.
from one of these dead sponges, or?
Oh. So sorry, Rennie, you were saying?
Yeah, we're gonna keep an eye out for rocks.
I see many a rock, though.
Mm-hmm.
So what type of rock are we looking for?
Whatever Beth wants.
Roger.
Yeah, it doesn't have to be right here. It was more like at our next
Yeah, at our
pause.
next juncture.
Once everything's settled out, if it looks like it's a good spot.
Starting to see a few more coral types in a single area again. Something red in the middle of the screen. Is that a sea star?
Oh yeah, it looks like it might be a shrimp or something. Where's my that little guy?
Yep.
Yeah.
Yeah,
Yep,
it's
shrimp.
like a little shrimp. Some more corals. Herc is on the move.
We are flying.
Oh, yeah. Sorry, guys.
Oh no, it's fine.
Just get myself in a little better position, but we can also stop for rock at any moment.
On a bamboo coral right in front of us. Steve, do we have a good idea of how old some of these bamboo corals can get?
Yeah, I mean, some of them have been aged before. It's not easy to say, since there's some variation amongst the different genera within the bamboo corals on how fast they grow. And then, you know, there aren't many aging studies from this part of the ocean. You know, the aging studies I know of are more North Atlantic, Gulf of Mexico for bamboos. So, or in the West Coast, Pacific West Coast. So it's tough to say for these particular colonies we're observing here. it's not unusual for a very large branching colony to be upwards of a couple hundred years old. That's been measured. But yes, it's kind of case by case, taxon by taxon information.
Okay, thank you.
Yep.
Looks like one might have broken off here.
Yep, that one looks relatively freshly fallen. You can see that it's probably been denuded of tissue, grazed by predators potentially.
Steve, when we see some of the big sponges, I know in shallower water with divers, they've done studies to, experiments where they show the flow with dyed water. Have we done that with ROVs in deep water for some of these? Because I would imagine it's not, well, perhaps dependent on where it is and what the currents are, it's maybe not as productive of a flow.
Yeah.
Sorry, stand by.
I don't know of any specific instances. I know we have dyed corals before. We've dyed tube worms before to evaluate growth rates. I don't know of any studies that have done that on glass sponges. I imagine it would be very difficult because you have a surrounding environment, right? That is, it's a flow. It's a very strong flow compared to the flow that's induced by the sponge.
Jess, thanks for coming up to this feature. I don't think it's quite what I'm looking for.
Great.
In the faraway view, it looked like it was maybe more black, but up close, it looks more hard and gray.
Roger. Thank you.
Looks like it's got another Iridogorgia on it or something.
Yeah. Shades of black and gray down here.
Thanks for that answer, Steve.
I suppose if you wanted to do some sort of study like that, you would need to design a chamber to isolate the target from the surrounding seawater flow. So you could take a look at that a little bit more closely.
What's going on with that rock right in front? Is that just discoloration?
Which rock are you
The light and dark, just in the middle of the screen at the top.
This up here?
Yeah. Is it just shadow? Yeah, it's just discoloration.
Yeah, I think it's both that it's kind of flat and discolored. Not seeing a lot of highly altered basalts in this area. So it looks like on high pack, we're in an area that's maybe a little bit, getting a little flatter as we're moving up the slope. So the terrain might not be as steep.
Would you like to visit that flat area there? Or?
Oh, I'm just narrating. I'm not necessarily saying one way or the other. Just for folks who can't see that.
Are we going to be going up this side, or are we cutting over to this side?
We don't have a defined trajectory just at the moment. So you can see that Rennie is drawing some potential circles where we could go.
Yeah.
So just trying to think, what is our current heading here? Yep. Yeah, so we've kind of been lateraling along this part here.
Yeah, came up kind of the center here, lateraled over that.
Yeah, so if we could maybe, come up into that flatter spot.
Okay.
You see where that kind of like right angle is just above your cursor?
Yeah.
And then kind of we can move in that direction towards where it gets steeper again on the other side of that.
Sure, over here.
Yeah.
I'll just do a few moves at this. Is that 120?
Yeah, because this is a big old rubble field right in front of us.
Sure. 120.
Roger, 120.
Bridge nav. 1 step 30 meters, bearing 120. Thank you. Yeah, we could then come up a steeper portion at a right angle or at perpendicular to it. That might be exciting for the end of the day.
Sounds good.
So one of the fun things for me as a scientist getting to use a robot to explore the seafloor is the situational awareness of, how are we moving our vehicle in space over this terrain? And so requires a lot of different people looking at a lot of different angles. It's definitely kind of a orchestrated dance down here.
And is it just my imagination, or did Remy's last ship move? Did we feel that, or was that just my imagination?
I do hear something.
It's possible.
It felt fairly stable, and I just felt it a little bit more rocking, so I wasn't sure.
I don't know that I felt it before you said it, but now I feel like we're moving. So I don't know if I just didn't notice it or if I was being persuaded. So for those who are joining recently and wondering what we're doing, we are just outside of the southern boundary of the Papahanaumokuakea Marine National Monument at a seamount that was mapped earlier today with multi-beam bathymetry to understand what its topography looked like. And we picked a transit up the north-northwestern flank of the seamount to see what evidence of life is on this in this environment, as well as to collect samples of the underlying rocks to try to date the age of the seamount, as well as understand the minerals that precipitate on the rock surfaces over time. These seamounts are probably many tens of millions of years old. We're not quite sure how old they are, and that's why we want to collect these samples. And so we're currently moving upslope, and in the area we're in right now, it seems to be maybe a rubble field, some talus that's moved down from somewhere else, maybe some in situ material here as well, some of these bigger boulders. We see some evidence of various types of corals, also some pieces of dead sponge, like what you see in the bottom left hand of the upper left quadrant.
And if you've been following for a while, several folks have been wondering if it's possible to see the route that we're taking. And I believe right now in the bottom left quadrant, you can see a little bit or a bit of the track we've been on and kind of